Hi, my name is Anthony Bridgen, and I'm a PhD student at the Sainsbury Laboratory at the University of Cambridge. I'm a member of the LISA group, and I'm also a member of Gombelinkies College. And I'm here to talk to you a little bit about my research and what I do day to day. So, you may have noticed, if you've been outside recently, you may have been less so in the past 6 months, unfortunately, that plants have a huge diversity in their architecture and their forms. And there's two demonstrations of that on the slide here. On the left, we have Arabidopsis, which is a model organism. The one on the left has been exposed to mechanical stimulation to mimic wind, and the one on the right has not. And you can see that the one on the left is much shorter compared to the one on the right. And the reason for this is that the plant needs, if it's in high wind and it's shorter, it's much less likely to break and be damaged. So this is sort of an advantageous form for it to take. But we also see that in the wild. So on the right of the slide, you have two Scots pines. The one on the left of the image has been grown in a fairly hospitable environment, a park, etc., and has a fairly traditional sort of cone shape, which you expect of a tree. Whereas the one on the right hardly looks like a tree at all. It's much more stunted. All the branches are facing in one direction, and this is because it's grown in a far more harsh environment with high winds and much more exposure. And this form means essentially that it's less likely to be toppled and to die. So plants need to be able to do this because they are exposed to a huge range of different environments, and they're unable to just up sticks and move because they have roots, they have a whole system, they're not motile like animals. And the reason that they're able to do this is due to their developmental processes. So we can think of plants as being composed a bit like Lego, blocks on top of blocks. But instead of a Lego block, we have a subunit called a phytoma. So a phytoma is essentially a small piece of stem with a subtending leaf and a little axillary meristem nestled in between. So a meristem is essentially just a point of rapidly dividing cells, so it's a growth point for the plant. And these axillary meristems can either be dormant or active, depending on the environment, depending on light conditions, nutritional status, predation, wind, water status, all those different things that a plant needs to account for, and adapt its shape accordingly. And the thing that controls this, whether they're active or whether they're dormant, appears to be a hormone called Auxin. So it was first noticed around 100 years ago that this phenomenon existed, known as apical dominance. This was noticed by, say, Sachs, who's sort of a giant in our field. And he noticed that if you took the top off a plant, so you took off the growing point, most plants, if you have a house plant, there's the top few leaves are where most of the growth is occurring. If you take that off, all the way down the stem, those dormant axillary meristems start to activate and grow out. And they do so from the top down, so in what we call a basipetal order. And he noticed that if you then took this apical section and popped it onto an agar block, essentially jelly, and left it for a day or two, and then just took that block by itself and placed it onto a decapitated stump, it was able to suppress the outgrowth of those axillary meristems. So he hypothesized that this apical meristem was producing a substance which then travelled down the plant and suppressed the auxiliary meristems from growing. And since then, a huge amount of work has been done in this area, and it's been shown clearly that this hormone is Auxin, but we're still working on the exact mechanism of how this is controlled. So this is sort of just to demonstrate that in sort of the hormone world of plants, a fun world, ABA, abscisic acid, gibberellic acid, and Auxin are considered to be sort of the most important hormones, whereas ethylene is sometimes left out and a bit ignored, which is sad, but it also is less important. So anyway, we'll move on. So there are two main hypotheses to explain this phenomenon of apical dominance. The first is the Auxin transport canalization theory, which is what I work on and my group works on. And the second is the second messenger hypothesis. So I'm going to talk to you a little bit about both of them. So the first is the Auxin transport canalization hypothesis. And this relies on the notion that in order for one of these axillary meristems to be able to grow out, it needs to be able to export auxin into the main stem, where most of the auxin transport is occurring. And that this auxin export requires a strong source of auxin, so the bud needs to be producing a lot, and it needs to have a strong sink for auxin, so there needs to be not as much auxin in the stem. So think a little bit of diffusion, which you've hopefully covered. If you have a high concentration of a molecule, it will tend to move to an area of low concentration. So it's a bit analogous to that. And it also relies on the fact that auxin export is self-reinforcing, i.e. The more auxin that is transported, the more that transport is reinforced. So there's a positive feedback loop. So I work essentially to try and gather evidence and persuade people that this is a correct hypothesis and there are valid points in it. So I try to change this man's mind. So in order to explain this to you a little bit more, I need to tell you about pins. And these are a protein which is found in the cell membrane of plants. And they act as auxin efflux exporters. So they ship auxin from the inside of the cell out of the cell. And these are necessary because of the form that auxin takes in the plant. So it exists as a molecule called IAA, indole acetic acid. And this can exist in two states, either a deprotonated, so without a hydrogen atom, or a protonated with a hydrogen atom attached. And in the apoplastic region, which is the region outside of the cell, the pH is low, so it's around 5. So in this region, the IAA is protonated, so there are free hydrogen ions which can bind to the IAA and make it into a neutral molecule, which can diffuse freely through the cell membrane into the cell, down a diffusion gradient. Because it's much harder for an ion to get through a membrane because it's charged, and there are other charged proteins and fats in the membrane which make that process hard. But when the auxin molecule enters the cell, the pH is a little bit higher, more like 7, and these molecules become deprotonated, so they lose their hydrogen ion and become a negatively charged ion, which is then it's much harder for that to escape from the cell. So it needs active exporters which use ATP to ship that auxin out of the cell using energy. And the existence of these proteins creates an opportunity for plants to be able to essentially direct the flow of auxin. So by placing those transporters preferentially on one membrane on one side of the cell, they can direct the flow of auxin where they want it and where they wish it to go and where it needs to go. So this is how you are able to have mass transport of auxin. So within the main stem of a plant, you have around the vasculature, so where all the water and sugar is transported, you have this thing called the PATS, which is the polar auxin transport system, which is essentially channels of cells where there's these pin proteins are polarly localized on the base of those cell membranes, and you essentially have this flow, this bulk flow of auxin down the stem. So I'm just going to talk you through a little bit of evidence to support the auxin canalization hypothesis and make us feel a bit more confident that there's some some good ideas behind it. So the first piece of evidence is on the left, which is from a paper by Sauer et al. 2006. This is a section through a stem, through the vasculature of a stem, which is expressing pin proteins which have had a fluorescent protein tagged onto the end of it, so we can see them. So in the box at the top, the first panel, this has had no treatment, so nothing has been done to it, it's just been cut and we've looked at it. The one below that, in the second panel, a supply of synthetic auxin has been placed on the right-hand side, so creating an area of high concentration and an area of low concentration, a strong source and a strong sink. And they noticed that the pin proteins are able to polarize in response to this. So they move from sort of being more localized to the basal membrane to one side, i.e. away from the source of high concentration. And then in the middle panel, we can see that this is taken from a paper by Balla et al., where they've taken a junction from where the main stem meets an axillary meristem, and they've sectioned through that, again with these fluorescently tagged pin proteins being expressed. And they've shown that when they've decapitated the main stem, so when we've got this release of apical dominance, you can see that the pin proteins are being sort of localized from the bud into the main stem, creating sort of a channel of cells expressing basally localized pin, indicating that this bud is able to export auxin into the main stem, which is one of the things that we think is key to it activating. And then on the right here, the last thing we're going to talk about is taken from a paper by Mazur et al. 2020. It's essentially showing that if you damage the vasculature, so that bit where there's there's transport of water, sugars, etc. If you damage that by making a cut and then supplying auxin above that cut, you see a generation of a file of cells expressing basally localized pin 1 from that source of auxin joining into the existing vasculature, suggesting that the polar localization of these pins is able to sort of format the induction of new vasculature, which again would be key if a new bud wants to export auxin into the polar auxin transport stream. It needs to be able to generate that vasculature through which it can export auxin. So, there's a reasonable amount of evidence to support this theory and suggest that at least some aspects of it are true. And we work to progress that further. And the other one is the second messenger hypothesis. So this works on the basis that auxin doesn't directly influence bud activity, which we know is true because through radiolabeling experiments, auxin doesn't ever actually enter the bud. It only travels through the main stem if it's being supplied from the top. So the hypothesis is that auxin is actually regulating two other hormones and the production of these hormones called cytokinin and strigolactin. So it's just that because auxin is able to suppress the production of cytokinin, which is a bud activator, and is able to activate the production of strigolactin, which is a bud inhibitor, that through doing this, it can modulate bud activity. So there are a few pieces of evidence for this. The first being that auxin is able to control the expression of the biosynthetic enzymes of these hormones. So if you remove the apex, the apical meristem, you can see that cytokinin biosynthetic enzymes, so these IPTs, are upregulated, so there should be more cytokinin being produced. Whereas strigolactin, it's actually the enzymes which produce this are downregulated, so we should have less strigolactin. There should be a release of inhibition of these buds and they should grow. And we can also see from applying these hormones directly to the bud, so if we apply cytokinin directly to a dormant bud, you can see it will grow out, whereas if you apply strigolactin to a bud where the apical meristem has been removed, it is able to maintain suppression of that bud and keep it dormant. So there's just a couple of examples of that here. So in the top panel, you can see what we call a Mitchison plot. So this is essentially a comparison of where you have two buds on a piece of stem and you take off the top, which of those buds grow out and which win. So you can see that if we supply nothing, if we just have a control experiment, it tends to be fairly even towards which of those buds grow out, so there's a degree of competition between them. But if we supply GR24, which is a stragolactone, a synthetic stragolactone, you can see that it tends to only one of these buds grow out, and only one tends to win. And that's because there's a competition for access, we think, to the main polar auxin transport stream, so that main channel of auxin transport. And we think if there's multiple buds on this stem, they're all competing to try and access that sink and develop vasculature into it. And of course, as soon as one of those wins, it's going to start exporting a lot of auxin into this stream, which means that that polar auxin transport stream is no longer a good sink for auxin, and the other buds can't access it as easily. And in the lower panel here, this is just a demonstration using a sort of a model system where we have a piece of stem with a single node or single bud on it. And if we decapitate that and supply auxin, but also supply VA, which is a synthetic cytokinin, you can see that those buds where that's been done grow out much more quickly and to a larger size. So it suggests that there certainly is an important role of stragolactone and cytokinin in controlling bud activity. But whether that's the only aspect is unclear. So in science, we have these hypotheses, and everyone thinks that their hypothesis is right. And that someone has to be wrong in order for someone to be right. But that's not always true. Sometimes little pieces of different hypotheses can be correct, and we form a new hypothesis. So it actually seems like, indeed, according to the Auxin canalization hypothesis, it's important that buds need to be able to export auxin into the main stem in order to activate. But it also seems like the ability to do this export using the pin proteins is modulated by stragolactone and cytokinin. So let me show you a little bit of evidence for that. So this is taken from Crawford et al. 2010. So this is essentially a demonstration that stragolactone, which is the one that inhibits branching, so it inhibits those axillary meristems from growing out, is able to essentially act to remove pin 1 or induce removal of pin 1 from the cell membrane. So if we're looking at panel A here, on the left-hand side, these are just sections through stems, again, expressing pin 1 with a fluorescent protein tag. If we look on the far left of that panel A, we have WT, which stands for wild type, and we have the top one, which is a control, so that's just been sectioned. And the one below that has been treated with GR24, which is that synthetic stragolactone. And you can see in those images, there are these sort of green, horizontal, quite intense lines, and that's polar pin 1. And you can see where the stragolactone has been applied, there are much fewer of those, so we can't see as many. And that suggests that GR24 is acting to deplete these pins from the membrane. And you can also see that on the far right of that panel as a mutant background called MAX2, which is a mutant which is unable to synthesize its own stragolactone. So we'd expect to still see a response there because we're adding our own stragolactone, so it should have the same effect. But the middle panel is a MAX1 background. So this mutant is unable to sense stragolactone. So even if we're adding stragolactone, it shouldn't be able to respond to that. And you can see, even when we're applying GR24 in that middle image, there isn't really a change in the amount of polar pin 1 on the basal membrane. So that suggests that stragolactone is acting to reduce the ability of pin 1 to polarize, which means that it's much harder for buds to develop that export of auxin because it needs the polar pin 1s to export the auxin. So that's one way by which stragolactone is able to control bud outgrowth and actually inhibit buds from growing by stopping auxin export. And so you can see there the two sort of aspects of the two hypotheses coming together, which is quite interesting. Um, so just move on. So, and we also see this in cytokinin. So from a paper by Waldy and Liza in 2018, so from my group, which shows that cytokinin is actually able to do the opposite. So where you apply a cytokinin, it actually increases the amount of pins on the membrane, and that this seems to not be a transcriptional effect. So it's not because there's more of the protein being produced, it's because more of that protein is being allocated to the membrane. So you can see here how cytokinin could act to help those buds develop export of auxin into the mainstream and therefore activate. And we've also shown by a paper by Bennett et al. 2016 that pins are responsive to auxin. So they're able to respond to their ability to transport auxin, if that makes sense. So if you remove a source of auxin, so this is data taken from stem sections again. So you have a small section of stem, you cut through it, you have fluorescently labelled pins, and you have obviously the top of that stem which would have been the more apical region, and the bottom of that stem which would have been the less apical region. And we can see that if we just take that stem and we just cut through it, after a few days, you don't see as much polarly localized pin 1. So that's in panel B there. You just see sort of smears of green, there's no real concentrated horizontal lines, which is what we're looking for. If we continue to supply auxin, of course, auxin is being transported, so we still see those horizontal green lines, we still see polar pin 1, which makes sense because we suspect that if auxin is being transported, it reinforces that polar localization and keeps those horizontal lines there. But actually, if we add something called NPA, which inhibits those auxin efflux transporters, those pins, we can see that it's able to maintain the pins on the membrane still. So that's suggesting to us that the polarity of the pins, their location on the basal membrane, depends not on the flux of auxin across the membrane, but on the concentration differential, because that MPA is trapping auxin inside that region of the cell. So just to kind of summarize what we've been talking about, is that in general, the overall architecture of a plant depends on the activity of these axillary meristems and whether they lie dormant or they're active. And that in turn is controlled by auxin transport, which regulates other hormones, and that sort of acts together to integrate all of these different environmental stimuli and mean that the plant is able to form an adaptive response. And this creates a sort of highly tunable system which can create the diversity of forms which we see when we look out of our window. But the real question is, why do we care? Why are we doing this research? So I want to talk to you a little bit about food security. So that essentially is the concept that everyone in the world should have access to a secure supply of food. And that should be healthy and nutritious food. And that is hugely under threat. In the next few years, our population is growing, going to continue to grow to around 10 billion by, I think, 2050. And already around 1 billion people are malnourished. So there's clearly going to be more issues here. And there are a few ways which we can tackle this. On a sort of a supply side and a demand side. So one is to reduce waste, because there's a huge amount of waste in our food systems, mostly actually at the consumer level. So, we all tend to buy quite a lot of food, we're like, oh, that looks nice, I'll have that. But then it sits in the fridge, often goes off after a few days because we don't get around to eating it and we chuck it. So we can, try and develop tactics to mitigate that waste, or we can increase the production of food. and there are a number of ways that can be done. So through applying pesticides, but they have their own issues because they can kill higher tier organisms in the food chain. Applying fertilizers, which we do in huge quantities, but they cause environmental degradation. We can attempt to do it through breeding processes, so fairly traditional, this plant's really produced a lot, and this other plant produced a lot, so if we cross them, this one should produce even more, that kind of approach. And sort of the final, and the one that has most potential in this context, is genetic engineering. So to understand pathways and processes in a plant and then manipulate them with a sort of a more pointed accuracy and try and make progress there. So I'll just sort of talk about two cases where this has been helpful in a sort of a breeding context. So on the left we have a sort of ancestral plant called teosinte, which is a close relative of what we now grow as maize or corn. And you can see here from this figure by Yang, 2019, teosinte is a very branchy, bushy plant with very small, not very tasty ears, whereas maize is a single branch producing quite large, sort of juicy ears. And the reason that there is this difference is because of an allele called TB1, and that was selected for in our crop maize over many, many years, thousands of years. And that's actually able to repress the growth of these axillary meristems. So we end up with a single stem, and a lot more of the energy, of growth energy, is directed into actual edible parts. So that's very helpful. And then in this right panel here, we have wheat. And this was a development made by a chap called Norman Borlaug, who was hugely significant in something called the Green Revolution in the last century. And he bred wheat to become increasingly shorter, which means it's less likely to topple over, which is one way we lose crop. And it's also directing more energy again into those edible parts, the ones we like to eat. And that was found to be regulated by an RHT allele, which in this case was not related to auxin, but reduced responsiveness to gibberellic acid, which is in one part regulates the height of the plant. So clearly these were found by breeding approaches, but imagine if we'd already known about those pathways, we could have gone, aha, if we just modify the TB1 allele or modify the RHT allele, we can instantly achieve these changes and increase yields. So, there's huge potential there. I'd just like to thank you all for listening. Sorry if I've rambled on a bit. I hope it's been interesting. If you do have any questions, please feel free to get in touch. I've put my Twitter handle and my institutional email address on the first slide. So, yeah, feel free to whip me any questions. Thank you so much for listening. Bye-bye.